This video is going to show how to analyze a trio according to an autosomal recessive inheritance pattern suspicion. For this, we are going to use the third example of the RD-Connect cases that can be downloaded from the test instance. Here, the index case is a 16-year-old female referred for muscular dystrophy and presenting with muscle weakness, dystrophic muscle biopsy, quadriceps muscle atrophy, and myalgia. As you can see in the pedigree, she has an affected brother, and both parents are unaffected, leading to an autosomal recessive inheritance suspicion. The first step, once you access the platform, is to enter the IDs of the different samples. Here we'll analyze a trio, and therefore enter three different samples: the index case, 3C, the mother, 3M, and the father, 3F. As we enter the samples, a phenotypes icon will appear to link out to the RD-Connect phenotypes instance and the detailed phenotypic information of the family. Also, remember that you can have access to a summary of this information at the bottom of the page. The next step consists in defining the inheritance pattern we want to assess. We will start looking for homozygous alternative variants in the index case and heterozygous alternative variants in both parents. This is in agreement with our suspicion of an autosomal recessive inheritance. On the right part of the screen, you will be able to define different quality parameters, such as the minimum depth of coverage and the genotype quality, set to 10 and 30 by default. The minimum and maximum alternative allele frequency will vary depending on the selected genotype, but you can also manually modify them. In this example, we'll use the default parameters. Now we can run the query to see how many variants are homozygous in the index case and carried by both parents. Results retrieve more than 200,000 variants. You will still need to apply other filters to narrow your query. The first filter option concerns the variant type. Here you will be able to filter by the impact that the variant has at the protein level according to SNP. We will select high and moderate. High refers to variants that have a truncating effect at the protein level, as you can see in the description hovering the mouse. Moderate are the variants assumed to affect protein sequence, such as non-synonymous substitutions. We will also narrow our search to protein coding variants. Next filtering step that we are going to use in this example is the population frequency. Here you will be able to exclude common variants according to different databases such as Exact, Nomad, or the RD-Connect database. For this example, we'll filter variants present in more than 2% of the Nomad population. We'll then try to narrow our query filtering by an in silico gene list panel. For this, we'll use the Gene Disorder and Phenotype tab and browse in the RD-Connect predefined gene list. You can see a small description of each list hovering your mouse on top of them. In this case, the patient presented with a muscular dystrophy, so we'll start to filter by the muscle gene table comprising more than 400 genes associated to muscle disorders. Now we can rerun the query and see how many variants we get applying this filter. This is how the results are shown in the platform. 2 variants passed our filters. The section starts with the chromosome and genomic position, linked to dd-SNP, the reference and the alternative allele, and the genotype of the individuals from our query. Then you'll have the information about the gene, the impact on the protein, the in silico predictors, and the population databases. In this case, we can see that this second variant has already been annotated in ClinVar as benign unlikely benign. So we'll be more interested in the clinical significance of the first one. If we select the variant and have a look on the top section, we'll have more information concerning its annotation: functional effect, results of in silico predictors, detailed population frequencies, interaction, and pathways. For data interpretation, we can also link to different databases at the gene level. For example, here we can link out to OMIN. We can see that this gene has been associated to Machado-Joseph disease, an autosomal dominant disorder. Therefore, this might not be the causative variant in this case. So what we would do now is to go back to our filters and change our genotype pattern to look for a component heterozygous. As we have a trio, We just have to select the component heterozygous box and rerun the query. Here we also retrieve two variants, both annotated as pathogenic or likely pathogenic in ClinVar. From there, we can link out to retrieve more information about the pathogenicity. Again, we can work through the different top tabs to help on the interpretation of the data. Finally, if we also link out to the OMIM database, we will see that this gene has been associated to a muscular dystrophy, which would explain the phenotype of our patient.